information on human genomic, environmental, and trait data. He's a pioneer in genome sequencing, and it's really exciting to note that George's work led to the first commercial genome sequence in 1994. He's got lots of exciting things to talk to us about, but this evening he's going to share an intriguing topic with us: genetic superpowers, changing your genome and environment. Please welcome George Church.
So this is my thank you slide and my conflict of interest slide. I'm very grateful for these. My biggest conflict of interest is, of course, my wife, who's not only a top-rate, cutting-edge research geneticist, tenured professor at Harvard, but also has this impact worldwide by helping to educate. And you'll see as I go forward, there's a lot of education to be done, and she handles it from high school all the way through physicians. And if you get interested, go to pged.org, and you can put a little red dot on your favorite part of the world by answering some questions. Now part of the reason we need this education so urgently is that the landscape has changed so quickly. It was on a Moore's Law curve. It's kind of this dotted exponential, the way that computers are changing exponentially. And it dropped off that curve in a very delightful way, so that the $3 billion genome came down to roughly $1,000 wholesale genome today, not in 6 decades, which we would have predicted if it went at the high rate of computers, but in 6 years. And we're barely ready for it. But part of being ready for it is our ability to share among researchers and the world the information about our genes, environments, and traits. And so we have this very special project that you've already heard about, personalgenomes.org, which is the world's only open access source for this information. And the idea is not just to predict from your genome, the exact hour of your death, but to fill in a lot of other information that will constitute precision medicine. It will constitute this big data set about each of us instead of the very small ones that we have right now. And the million-fold improvement in the cost and quality is not only reflected in your inherited genome, but in the genomes of your microorganisms, your viruses, allergens, your immune response to them, and so on. Some of the people in this project decide to become re-identified. They share their name. It's not required, and they show up for an annual meeting where we bring together scientists, engineers, clinicians, and the volunteers. It's a unique and wonderful experience every year on DNA Day. And what we do for them and with them, and it's a very participatory project, is we measure everything we can imagine, and that list of things we measure is getting larger and it's highly integrative. So for example, we measure everything from their genome to their stem cells. We can differentiate their stem cells into brain-like structures and analyze the actual brain in the functional magnetic resonance imaging image there. So that's actually my brain.